Thank you all for joining today. It's really my great pleasure, real pleasure to have my friend Dr. Guillem Pratx from Stanford University. And Guillem and I we experimented together when we were postdoc, and he's always been the brightest, also the tallest guy in the room. And we have some really good time together. Um, so Guillem got PhD in actually in electrical engineering. So he's the engineer by training. He did a lot of work on optics, and recently he has been doing a lot of new stuff like single cell whole body PET, radioluminescence microscopy, flash therapy. There's many things you may really find very interested. So after his PhD, he actually did a postdoc in radiation oncology, and he stays as a tenure track faculty in the department. Now he's a tenured professor at Stanford. He has published some really good work, a lot of Nature papers, and also he is heavily funded by NIH. He's running like a 3 R01 active R01s, and he has been winning a lot of awards. I don't want to, talk everything, but I will give the floor to Guillem, and please join me and welcome the talk.
So thank you, Sean, for the very kind words, and for the opportunity to visit today. Enjoy the visit so far. Working around Irvine, first time to be here, and I look forward, meeting some of you later this afternoon. So I just wanted to give a kind of a snapshot of research going on in my lab. I'm a physicist working in radiation oncology, but I don't work with patients directly. I mainly do research, and by training, I come from a background in positron emission tomography, X-ray excited luminescence, and different ways of using physics in cancer for research and for diagnosis and treatment. So today, kind of the angle I chose is to come present from the angle of these microphysiological models. So these are very small models that are built in the lab that we can use to try to simulate complex clinical oncology workflows, right? And so what do we mean by workflows? Oncology is a complex field, and there's just many different things that happen in oncology, different treatments, different tools for diagnosis, imaging, staging, and so on. Of course, you can model each of these individually, and I won't talk about all of that. I will focus on work we've done in modeling imaging that happens in oncology, and also modeling radiation. So you can see those two. These will kind of be the two parts of my talk today. Will be using these models for imaging oncology, imaging and therapy. And so, what are these 3D microphysiological tumor models? So this is kind of a kind of a broad class of models that are typically grown in the lab and can be seen as alternative to animals, right? And so these include, sort of simple spheroids, which are just assembly of cells that are grown kind of in a compact fashion, where this allows for the formation of gradients in oxygen, nutrients, enables signaling between cells, to organoids, which are a bit more complex since these can be derived from cells that are directly taken from patients, and they're grown so that the stem cells will differentiate into different lineages of cells using special media, using 3D matrix that reproduces the extracellular matrix of tumors, and then tumor on a chip, which is adding additional functionality using microfluidic devices, which are those tiny fluidics channels that can be used to manipulate cells and reagents, simulate flow, simulate shear stress, model the vasculature. So this kind of a broad category of models that's encompassed by this term MPS. And recent, I guess this year, there's been announcement regarding new priorities at the NIH, trying to maybe move away from animal models in some cases and trying to leverage these new technologies that leverage human cells and use technology to replace animal models. And the FDA and the NIH have both sort of made announcements in this direction. And the reason for that, I think, one of the reasons is if you look at the just attitudes of the general public towards animal research, you can see in the graph on the right is that we're now about almost 50% split, where basically half the people have a negative attitude towards animal research and think it shouldn't be done, right? And so I think that's maybe where this is coming. Of course, animal models can do many more things than these simpler models can. But at the same time, animals are not humans, right? And so a lot of the animal models are not predictive of clinical trial endpoints. And so, so 3D cell cultures, so there's a big difference between growing cells like in 2D in a dish and then growing them in 3D, where it really recapitulates more of the architecture of the tissue that we see in tumors. And so that's a essential part of these models is to grow cells not in 2D on the surface, but in a three-dimensional matrix. And then the other thing too is when these models are grown from patients, from tissue that's derived from patients, these models can often predict the response of individual patients. So here's one example where patients basically had surgery and they were also treated with chemotherapy. And some of the patients responded to chemo, the other did not, right? And then by looking at the growth of organoids that were grown from this patient, you can see there's a significant difference in growth rate. So really the model recapitulates and kind of predicts what happens in the patient from which the tissue were derived. And so it's almost having a virtual twin of the patient, but in the dish where you can do multiple experiments. But so the goal today and what I will talk about is to actually look at not therapy, but looking at imaging, right? And my lab has worked with PET imaging for many years. And what we were trying to do, and we eventually did, was to develop a microscope compatible with PET tracers to image these novel 3D tumor models, right? So these tumor models, they're tiny, they grow in a little dish or a little chip. And typically the way they're imaged is through microscopy, because it's the tool that's best adapted for imaging these tiny models. The thing is, microscopy gives you endpoints not really relevant to the clinic, because in the clinic, what do? We get CT scans, MR, and PET, right? So what we wanted to do is try to see if we can add PET to our microscope so we could image these models using PET tracer. So positron emission tomography in the clinic and in oncology, it's really the gold standard for looking at biology and specifically looking at molecular processes non-invasively. And typically the way this is done is using a tracer. So it's a molecule that's radiolabeled. It's injected and it will accumulate based on some molecular process, some enzymatic process in different parts of the body and ideally in the tumor. And that's a quantitative readout of biology, right? So a PET scan gives you a number that reflects really the underlying metabolism in some cases of the tumor or the expression of a marker. And it's kind of a number you can use to make decisions. And, so just kind of like maybe an hour from here at UCLA, the first preclinical PET scanner was built in 1997. So the vision at that point was that people were doing PET scans in the clinic, and they needed a way to do this in small animals so they could sort of mimic the oncology workflow, right? And the idea was to build a scanner that's scaled down, that has higher spatial resolution, higher sensitivity, that basically can provide similar images in small animal models. And nowadays, I know you have one of the successor of this system here, and most universities have small animal PET scanners. It's widely adopted, and it's very useful to be able to do the same clinical endpoint in small animal that you would do in the clinic. And so now we wanted to look at these other models, which are much smaller than animals, and for which a PET scanner is just, the resolution is not good enough. And so we developed this radioluminescence microscope, also known as RLM. And I know this is a optics institute, and so I won't get too much into the detail of the optics, but the way this works is we have a microscopy objective here, which is pretty standard. It's an infinity corrected objective. And what we do is here replace the tube length with something that has a shorter focal length so that we can kind of demagnify image. And the reason for that is we're trying to measure these very dim events. So whenever a positron decay occurs, it will emit a little bit of light through a scintillator that we're trying to capture. So mainly what we're trying to do here is capture a little flash of light each time there's a radioactive decay that happens. And so to do that, we need to get as many photons as possible on the smallest number of pixels as possible. So we demagnify the image, and that's how we do it. This is kind of the simplified version of the workflow. There's a glass slide on the bottom. We have cells that are plated on the glass slide or inside a dish. Then a radio-labeled molecule is introduced. That could be, for instance, FDG, PET tracer. And then we bring in a scintillator, which is kind of like a cover slip if you think about it. And whenever the radioactivity decays, you can see the positron is basically traveling upwards into the scintillator. And you get a little flash of light when that happens, right? And so there's numerous scintillator materials we can use for this purpose. So these are some images just to first look at resolution. These are two cells separated by 22 macrons. This is an image of the distribution of FDG, and you can see we can resolve the separation between these two cells. The halo you see around is because the positron, they're not like photons. They don't travel straight. They tend to scatter. There's a halo around the two cells, but we're able to see kind of there's two single cells here in this dish, right? And then the microscope we also have, because the scintillator is essentially transparent, so we can just do standard fluorescence imaging as well. So you can see on the top, this is just a nuclear stain. We can also do bioluminescence. So because microscope is very sensitive, so even like something very faint like bioluminescence, we can image. And then FDG uptake, and then we can combine all that to make an overlay. There's some reconstruction that goes in also. So there's two different ways to make images. So this is what we do. We call digital is actually counting individual pulses that are coming into the camera and then making an image out of that. Or analog is just essentially integrating on the camera all the lights that's coming out. So it gives you an image that's a little bit more blurry, you can see here, right? So analog is more blurry, and then digital is much sharper and more quantitative image. So now let's move on to 3D microphysiological models, right? So I'll show you just 2D cells and what happened when we move on to 3D. One of the main effects when we move to 3D is the effect of depth on sensitivity and resolution. And so this is an example where we had sources, for simulation, but sources placed at different distance from the scintillator. And what you can see is that as the distance increases, the resolution degrades. At first slowly, but then eventually, we essentially lose the sensitivity, and it's because there's too much diffusion. So we can, based on this, we can go back maybe 40 macrons deep on these models. So that's before the resolution is degraded. And so then, we can look at different types of models. So I showed you some cell cultures already, but we have spheroids, organoids, tissue explants, and then the tumors on the chip. And we've worked with all these models, so we've actually have experience with those. So what does the spheroid look like on the microscope? So this is an example with A549 cells, the lung cancer cells. And these were grown in, so essentially cancer cell, they grow in the low attachment plate, so they form a little clump of cells. It's about 20,000 cells. And so you can see in the spheroid, because of the gradients in oxygen and nutrients, the core of the spheroid often is going to be made of dead cells that are starved of nutrients or starved of oxygen. The core is also hypoxic, as you can see on the second image. And then when FDG is introduced, it's going to be taken up mainly on the rim of the spheroid. First of all, because of the diffusion effect, it has to diffuse all the way to the center, and it gets consumed along the way. The other reason is because simply the cells in the center are not, they're basically metabolically dead, so they're not taking up FDG. But we can see some resolution of that effect, right? The rim of the spheroid is visible. And then so we were interested in trying to move on to organoids that are derived from tissue samples from the clinic. So we worked with a surgeon who operates primarily on head and neck tumors. So these are like squamous cell carcinomas. And there's a lot of work that goes into the logistics, being able to collect the tissue when it comes out of the OR. And what we do is to collect the tissue, mince it into very small chunks, and then it's basically cultured in it's called a matrigel dome, which is this sort of concave shape. Essentially, it's a matrix that supports the growth of these organoids, that provides nutrients. And then there's a special media that contains many growth factors and inhibitors necessary for these to develop into full-grown organoids that really recapitulate the in vivo tumors. And so these organoids, then we take them out of the matrigel and we place them on the scintillator, and we're able to acquire images of them using the microscope. And so this image you see here, the reason why the field of view is truncated is because we're demagnifying. So we're actually losing some of the field of view because of demagnification. The sensor of the camera is basically too big for the image we're looking at. So I think I just showed that one. Oh, so there's two different ways to do organoid culture. There's the matrigel dome, which is at the bottom of the media plate. And then this air liquid interface, which is in the transwell plate, which is, I guess, helps preserve integrated stroma. So this preserves more of the immune cells and fibroblasts. And the reason being is the oxygen can penetrate more easily into that setup, and it's more supportive of the growth of these organoids. And so, working with other, so Johnson, who is the surgeon, and then Calvin Kuo is the organoid expert at Stanford. So we just imaged a number of different organoids, right, derived from patients. And so you can see you don't always get like a signal out of each organoid. So some of them you can see there's not really any uptake that's detectable. Some of them have higher uptake or lower uptake. So there's some heterogeneity in these organoids. And so just to give an example of whether there's correlation, because I think that the key question is whether the signal we're seeing in the dish correlates to what we see on the PET scan. So we've actually done is for each patient that goes through surgery and donates tissue, we also get the PET CT for these patients. And this patient was interesting because it's actually a thyroid cancer patient. And thyroid cancer can be interesting in the sense that it can be more differentiated than in that case, there's low FDG uptake, or more or de-differentiated, and then there's higher FDG uptake. And this patient had two thyroid lesions that were removed. And one of them had a very high FDG uptake of 32, whereas the other one was barely any detectable uptake, right? So we took both lesions. Both of them yielded organoids that were viable. But then, when we did the measurement with FDG, you really see again the reflection of the biology being different in those two lesions, with one lesion having very high uptake again, and the other one much lower uptake. So this is an interesting endpoint because we can look at this number we get from our assay, and it sort of correlates to what you would get from a PET scan of the patient. So we got more thyroid cancer organoids through a collaboration. And one other thing we were interested in is thyroid cancer is typically treated with radioiodine. Your thyroid requires iodine to be healthy, but thyroid cancer requires lots of iodine, and that can be used as a marker for diagnosis of thyroid cancer, also as a treatment pathway using radioiodine as a therapy. And so all we did here was just to verify that these thyroid organoids also maintain functional radioiodine transport. So if we give a radioiodine to these organoids, we're able to show they still maintain uptake of radioiodine. So they still really reflect the behavior of thyroid cancer in the clinic. And then so another class of models that's a bit more advanced is the so tumor on a chip or tumor chip. So these are built inside microfluidic devices. So microfluidic devices, this is made of PDMS using microfabrication techniques. And this is a very common type of, see this. So I guess it's reflective, but so it's a very common type of chamber for culturing cells in 3D. And you can see there's two side channels, the red one and the blue one. The idea is to grow the tissue in between those two channels in 3D, and the two channels on the side are meant to be essentially vasculature, right? And so in this case, what we do is we induce hypoxia in one of the channel and then have normal oxygen on the other channel, which creates a gradient of hypoxia throughout the whole chamber. And the other thing we did is we able to couple the scintillator directly into the macrophilic device so that it's an integrated system that has the macro channels and the scintillators all in one system, so that we can sense radioactivity in the device, take images of it, and we can culture these cells. We can also treat the device using X-ray, so kind of to simulate radiotherapy. And so looking at these channels, the cells are grown in the central channel here. We verify that there's induction of hypoxia that's either uniform, so we can have normoxia, which is high oxygen throughout the whole channel, or hypoxia throughout the whole channel. So green means hypoxic here. Or we can have a gradient where one side is hypoxic, the other side is not. And there's basically most tumors are hypoxic, but heterogeneously hypoxic. There's always gradients of oxygen because oxygen diffuses from the vasculature. And so this really mimics, what tumor is supposed to look like. And so we did different assays, and here just to look at one of the assays. So why do we care about hypoxia? The reason hypoxia is important is because, and I'll kind of go back to that a bit later, hypoxia is really one of the main determinant of radiotherapy response. So in the absence of oxygen, radiation is much less effective at killing cancer cells. And it's just because on the chemical level, radiation requires oxygen to really damage DNA. And so because of that, you can see that in the absence of oxygen, so under hypoxia and giving 10 gray of radiation, there's much less DNA damage than in the presence of oxygen. And it's about a factor 3 fold dose enhancement with oxygen. So it's really significant. But then when we have a gradient, essentially, we also see a gradient of DNA damage. But the idea is that even if you have a small pocket of hypoxia in the tumor, this can really drive the recurrence of the tumor because, again, it's a 3-fold enhancement in dose needed to control the hypoxic cells. And so again, so like I mentioned, we have a scintillator, so you can actually see it here. The scintillator is this square here. This is the PDMS device with the macro channels here. And these are images with the radioluminescent microscope. You can see by the circle. And so, we're able to capture and look at FDG uptake as well in these devices. And the interesting thing here is that under hypoxia, actually, tumor cells upregulate the uptake of FDG. The reason for that is in the absence of oxygen, cells have less efficient metabolism because they use anaerobic glycolysis, and therefore they require more glucose. So they will upregulate glucose uptake in order to continue metabolizing. And so you can see a small increase and then, maybe this gradient that happens in FDG uptake. So, sort of the main goal here is basically we're trying to use PET as a way to have a bridge between the different models and then clinic, right? And so PET can be used in small animals. It can be used now in macrophysiological system, and it can also be used in humans. And so because it's quantitative, it is a biomarker that sort of spans the multiple scales, right? From tiny models all the way to humans, right? Which is, I think a way to make some of the research more relevant to the oncology workflows. And so RLM is a way to put high resolution PET into macrosystems. Of course, it's not PET in the sense that it's 2D. It's not 3-dimensional, right? And it's really kind of a 2D projection rather than a 3-dimensional tomographic image. But it is a quantitative method because we are counting individual decay. So we're getting numbers that are relatable to how much radioactivity is present in the cells. And it's really compatible with a wide range of in vitro models, whether these are like monolayer of cells or thicker three-dimensional models. Things that we were looking at, in terms of this project is we really want to put so real vasculature, right? So these are actually chips we've been growing in the labs recently, where we seed the chips with not only tumor cells, but endothelial cells, and they arrange in these networks. And these networks are perfusable, so we can actually perfuse them and really mimic, kind of like a real tumor that has a vasculature. And then we want to also add patient-derived organoids and combine the two and create these models that really are more reflective of in vivo tumors. So that was sort of the first part, looking at imaging. And now I also want to talk about some of the work we've done related to radiotherapy. So I talked a little bit about radiotherapy, but I'm going to go into more details. And specifically, I want to talk about flash radiotherapy and the flash effect. So I mean, I'll just give a bit of background, just because I know some of you may not be familiar with flash radiotherapy. So radiotherapy normally is use of X-ray or ionizing energy to destroy tumor cells. So flash radiotherapy is it's very similar to normal radiotherapy, except the radiation is delivered very fast, right? So it's called flash because it's essentially giving the radiation in a fraction of a second as opposed to several minutes. And as a physicist, there's not really any difference, right? You're giving the same amount of energy. It's the dose deposition is the same, dose distribution is the same. So it's really at the chemical and biological level that things are different. And what people call the flash effect is the fact that the finding that there's actually an increased therapeutic index when using ultra high dose radiation therapy compared to conventional dose rate irradiation. So this is a finding that was discovered about, I guess now it's about almost 10 years ago, over 10 years ago. But the finding was that when the same amount of radiation is given at different rates, there's actually a sparing enough more tissue that happens when the radiation is given as flash compared to the conventional dose rate. And so some of the examples for that are shown here. So this was the seminal paper in 2014 that showed that in the lung, giving radiation as flash radiotherapy causes less fibrosis. So you can see in that third column here. So it's the same dose, 17 gray radiation. You can see markers of fibrosis here, and here basically there's clearly less fibrosis that's shown. In the pig and the cat models, there's less skin damage. So you can see on the pig skin, clearly like less necrosis of the skin when radiation is given as flash. And there were trials done in cats that were showing better sparing of skin as well. So this is all good. And then there's additionally, so work done at Stanford looking at GI toxicity. So these are mice that receive abdomen irradiation. And actually, most mice will die when they get 16 gray to the abdomen because of severe GI syndrome. But with flash radiotherapy, most of the mice survived, right? Which is, really, really strong effect, right? It's and then flash can also preserve neurocognition. So these are functional experiments looking at mouse behavior after radiation with flash and conventional radiotherapy. And again, this shows that so radiation can basically adversely affect neurocognition, but flash can preserve neurocognition. So these were very exciting to the field because as radiation oncologists, the main issue is the side effects, right? Because we can always treat tumors, give enough radiation will destroy the tumor. There's no problem with that. But the side effect is really what's preventing us from curing more patients, right? But I think all that would be great, right? But what about tumors? I think that what made flash such an interesting topic is the effectiveness with tumors is the same. So flash radiotherapy spares normal tissue, but has exact same effectiveness for controlling tumors. I think this probably needs more research, right? But I think all the data so far, and there's been multiple studies in small animal, really suggest that there's no difference for tumor control, right? So there's different curves looking at tumor growth in mice, survival, and haven't really found a study yet that has shown a difference in terms of tumor control. And so many in the fields have really called flash radiotherapy the holy grail of radiotherapy. If we can treat patients and they don't have side effects, we can cure the tumors it's great. I mean, that's really what, there's nothing better to hope for, right? The problem with radiation is the therapy always comes with side effects, right? And so typically we look at normal tissue toxicity. So the blue curve here is as we increase the radiation dose, the probability of normal tissue toxicity goes up, right? But the probability of tumor control also goes up. And so it's always a trade-off, right? Like as an oncologist is how much normal tissue toxicity are we willing to tolerate to get a good probability of tumor control, right? And it's very narrow. I mean, the window is very narrow, so it's very hard to find the right dose and the right treatment. But with FLASH, we can push this normal tissue toxicity to the right and really widen the window. So what is different about this FLASH radiotherapy? Well, what it does is it's a very fast dose deposition. So what happens is there's a lot of chemistry that happens faster than the radiation can be deposed. It happens more slowly than the radiation itself. And so the radiation can be given faster than the diffusion of oxygen, for instance. So that means oxygen can be depleted, and new oxygen doesn't have time to replenish, right? The tumor, for instance. There could also be reaction between radicals. So radiation generates radicals, and these radicals can react with each other, which only happens if the radiation dose rate is high enough. It could also have effects on like circulating cells, right? So if cells are circulating and the radiation is given very quickly, most likely they won't have time to circulate through the field, and therefore they'll be spared. But we're mainly looking at oxygen effect, and that was sort of our interest because oxygen is really crucial to radiotherapy. So these are some clinical examples that, patients that have a hypoxic fraction greater than 50% tends to have worse survival. So radiation is less effective in these patients. So this is well known. It's shown clinically and also preclinically. And oxygen only needs to be there for milliseconds before radiation to have an effect. And the reason is that in the absence of oxygen, so radiation will create radicals in the DNA that can be restored by glutathione, which is a reducing molecule. But in the presence of oxygen, there's a competition between glutathione and oxygen. Oxygen is typically faster because it's much more reactive. And that gives rise to these radicals that are much harder to restore, at least chemically. The literature also has shown that the oxygen levels drop during FLASH RT. So there's been many attempts to measure oxygen levels in real time during FLASH radiotherapy, and including by us. And the yield, so the yield of oxygen depletion, people have reported different numbers, but everyone agrees there's depletion of oxygen. And in terms of chemistry, also it's fairly well understood how oxygen is depleted. Just to go quickly, water is broken up into radicals, and these radicals will react with oxygen, like through this secondary reaction, therefore depleting oxygen. And as well as lipids. So lipid radical will also react to oxygen through peroxidation reaction. And that also contributes to consuming oxygen. And this reaction happens fast enough that the oxygen level can actually drop during the radiation delivery. And the last thing is, so what about normal tissues? The drop in oxygen is not very large. It's relatively small. So really, this is something that's going to have an effect mainly on regions of the body that are already at low oxygen levels. And stem cell niches, a lot of them are hypoxic. And so the idea is that stem cells are really crucial to organs regeneration after radiotherapy. And so if these stem cells are preserved, the organ toxicity will be less, especially the longer term toxicity. And so some of the work we've done is trying to measure radiochemical oxygen depletion during FLASH radiotherapy. So we're using a phosphorescence quenching method. So this is a probe that's quenched by oxygen, which affects the lifetime, the decay lifetime of phosphorescence. And so this is an experiment. We had a pulse laser, we had the nanoprobe in water and irradiation with electrons. And basically the electrons reacting with the sample here, which was at a set oxygen concentration, depleted the oxygen, and we can measure that through a change in the lifetime of the probe. And so, these are sort of typical readouts we get where we start with, let's say 80 macromolar of oxygen. And as we deliver different doses, basically the oxygen level goes down like almost like in a stepwise manner. And so we're able to, by delivering radiation over and over, fully deplete the oxygen, right? So importantly, so both flash and conventional deplete oxygen. It's not just flash, but because flash is given very fast, there's no time for oxygen to be replenished from the vasculature. And so we also did some simulations. So this is kind of earlier work where we try to simulate what would happen if you had a tissue that had a single capillary. So it's called the Krog model, where oxygen diffuses out of the capillary, and then it's either depleted through reaction with radiation, it's metabolized, or continues to diffuse. And we come up with sort of predictions of what would the oxygen level look like in such a scenario. And so based on the length of the pulse of radiation that's given, whether it's 1 millisecond, 1 second, 10 seconds, you can see that if the pulse is 10 seconds, we don't really see much of a depletion because of the resupply. And below 1 second, we can start to see there's a significant depletion. And then 1 millisecond is basically zeroing all the way down. And interestingly, these were confirmed later by another paper that was published and actually measured depletion in vivo. And so you can see the time here is about 5 seconds for the recovery, and very similar to what we had predicted 3 years earlier. We also get about 5-second recovery time for oxygen. So yeah, I think it's I think these models kind of make sense. The main question I will come to, is sorry. So the other thing I wanted to mention is often measurements are just it's a average over a certain volume, right? So when we measure something, maybe it's 20, it's not like showing you exactly the distribution of oxygen. It's showing you the average within the interrogation region. And you may have something that looks like a small depletion on average, but really on the macroscopic level, there could be a region where it's higher because it's further away from the vasculature. So I think it's very complex to relate what we see in measurements and what happens really at the macroscopic level based on where the vasculature is distributed. So we did some simulations as well, trying to understand like the dynamics of depletion in these complex three-dimensional models. So this is built from true mass brain vasculature and looking at these equations. I think the point here is really, a lot of people have said, well, the depletion is not very much. Well, the reason for that is because it's bulk measurement. If we look at macroscopic measurement like this, and we wish we could measure this. I think this is very difficult, but if we could measure the macroscopically oxygen depletion, I think that would really answer some of these questions. But the simulation at least suggests that. And so now I just wanted to kind of briefly go to kind of what's a paradox, right? That in what I've said, I don't know if you were paying attention, you may have caught it, but basically the paradox, what about tumors, right? If we're depleting oxygen, why aren't we sparing tumors? Tumors are mostly hypoxic. We know that hypoxic tumors are protected from radiation. We're depleting oxygen. They should be even more resistant, right? But we don't see that in practice. So why aren't tumors more resistant to radiotherapy? We are depleting oxygen. It's been shown. We know that hypoxic tumors are more resistant. It's the logical implication is flash should not be effective in tumors. We should see sparing. I think that's really like the key question for me is why aren't we protecting tumors? And I have maybe the beginning of an explanation that I'll try to show you. So this is the way I understand it. So in normal tissue, when we apply flash, what happens is the depletion of an oxidant, oxygen. Therefore, there's less cell killing. So it's sort of shifting the balance towards radioprotection. In tumor, we don't see any radioprotection. So what's going on? Well, the hypothesis we have is that we are depleting oxygen. It is radioprotecting, but at the same time, we're also depleting antioxidants. And so this is why I kind of call the anti-flash effect. The idea is there are all these antioxidants in the tumor that can be depleted. So glutathione is an example. Radiation depletes oxygen, but also depletes antioxidants as well. It uses them. And so what evidence do we have for this? So here's some evidence. What happens if we remove oxygen and we give flash radiotherapy? You would predict that if you remove oxygen from the tissue and give radiotherapy, there would be no sparing compared to between flash and conventional, right? Because there's no oxygen to deplete. And so the data suggests, for multiple papers, suggests that oxygen is required to have a flash effect, and in the absence of oxygen, there is no flash effect. So these are multiple studies in different models that have shown that. So certainly oxygen is involved in the flash effect. Now, what if we remove oxygen in the tumor? What would happen? Well, so if we're removing oxygen in the tumor, then we would not see a flash effect. We would not see the radioprotection anymore, but we still have this anti-flash effect that doesn't depend on oxygen. So actually, if we remove oxygen in tumors, what we would see is an increase in tumor cell killing. Oh, and I think the scale at the bottom should be corrected. It's more cell killing at the bottom. And there's one study that was published in the Radiation Oncology journal that actually has done that. They clamped the tumors to remove oxygen. They treated with radiation, with flash and conventional. And what they found is actually, so with the clamp, this is conventional, and this is flash right there with the clamp. It's actually more effective to treat tumors with flash than conventional when the tumors are clamped. And so this also kind of supports this idea. And so, this is maybe a bit complicated, but the key difference between normal tissues and tumors is glycolysis, right? And glycolysis is really linked to this glutathione cycling. And so in tissue that are highly glycolytic, they're able to recycle glutathione really quickly because of the metabolism, and therefore they're kind of more susceptible to this depletion of antioxidant that I was mentioning. Yeah. So now let's go back to microphysiological models, and so we've been using spheroids as models for our studies of flash radiotherapy. We actually have a model that's based on lung cancer cells spheroids, and it's a very reproducible model. So these are 5 experiments we did, and the endpoint we look at is survival. So clonogenic survival of cells in these spheroids. Essentially, you get the same, we do the experiment 5 times, you get the same result 5 times. It's very reproducible. It's a very nice model. We either pull the spheroids when we do clonogenic survival, or we can also do clonogenic survival on individual spheroids. But it really, it's probably one of the only in vitro models of flash radiotherapy that's out there, but it's very convenient. One thing we were looking at is, in terms of this antioxidant depletion hypothesis, is trying to deplete antioxidant prior to flash radiotherapy. So we're using Erastin, which is a drug that inhibits glutathione synthesis. And the drug by itself is not toxic. It doesn't really affect survival in the absence of radiation. But in the presence of radiation, it is essentially a radiosensitizing drug because glutathione is crucial in protecting cells from radiation. And so what you can see is, with the blue curve for conventional radiotherapy, you can see an increased cell killing with Erastin. And flash, you see the sparing, but the sparing basically goes away as we increase the concentration of Erastin and we deplete glutathione. So again, I think it's maybe more evidence about really the role of this potential anti-flash effect. And also, we're also looking at DNA damage. So survival is one thing, but DNA damage is another, endpoint for radiotherapy. And here, we look at DNA damage at 72 hours. So this is really the DNA damage that hasn't been repaired. Most DNA damage will get repaired pretty quickly. But 72 hours this is the DNA damage that's going to kill the cell because it couldn't be repaired. And there is a small difference. So if we look at it between flash and conventional, but here again, glutathione depletion increases DNA damage. Again, it's due to the fact that glutathione is crucial to repair DNA damage. And flash lowers DNA damage. Therefore, sort of the same hypothesis that essentially it's causing less DNA damage conventional, and that's because it's depleting oxygen. So, it's both depleting oxygen and glutathione at the same time. So, yeah, so I think I will maybe wrap up now. And I think these are the crucial questions we must answer. First one is, what are the specific attributes of tumor that make them immune to flash radiotherapy? To me, I think it has to do with the metabolism and the fact that they're highly glycolytic. But then the following question is this attribute present in all tumors? So if patient come and they getting flash radiotherapy, is there a chance they may be the tumor may be protected against the radiation treatment, right? That's really a question we need to answer. And for that, we really want to be able to measure the presence of that attribute prior to flash RD in the clinic, because we may have to select patients who really going to benefit from this flash radiotherapy. And so, yeah, to wrap up, so I think flash it is promising. Again, I said it's potentially the holy grail, right? Being able to reduce toxicity, but there's these paradoxes that still need to be answered. So we know we can potentially spare normal tissue, which is really the number one thing that oncologists want to achieve is less side effects. We still don't really understand the mechanisms. Why? Is it oxygen? Is it radical reactions? And there's sort of this mystery of the fact that tumor control is achieved, right, despite oxygen depletion. So, so this is kind of closing. So this is from the same movie as my first slide, but as it said, it's so you must choose, but choose wisely. As the true grail will bring you life, a false one will take it from you. So, in terms of a patient, we want to be sure that we're bringing them the true grail, not a false one that's going to be harmful or, spare the tumors and lead to worse outcomes for them. So, conclusion, I want to thank you all, being here, having me here today. This is my lab who has contributed to this work. We're happy to be funded by NIH right now. We have many collaborators. So flash radiotherapy, the way we give it is, really a team effort. We have people who work on the beamline and able to deliver radiotherapy, people working with models, computing, and so on. So it's, again, pleasure to be here. And I'll be happy to take any questions you might have. Yeah, thank you. Yeah.
Thank you so much for really wanting to talk today. I have a question, so
Maybe.
Oh, yeah.
Thank you so much for the really great talk today. I had a question regarding flash radiotherapy specifically. One of the big topics with it is that it's already being translated into through clinical studies, but it's not shown in multi-institutional studies that the flash effect is always present in the same models with animals. Do you think that this has to do with that difference in oxygenation of the sites being chosen for those studies, or is there other criteria that ensure the flash effect occurs?
Yeah, I think it's a great question, and thank you for asking it. It's I think the flash effect is a bit finicky, and you're right. Some places have reported they did not see a flash effect, right? There was a paper recently showing that the choice of anesthesia, for instance, can influence whether there is a flash effect or not. So people who are using gas-based anesthesia, which typically is 100% oxygen, I've shown that they don't see as much of a flash effect because they're basically modulating oxygen by giving anesthesia, whereas other groups of using injectables, they see a stronger flash effect. There's also even sometimes the same group will see a flash effect on one type of beamline and not a flash effect on a different beamline, and these are presumably people who are using the same models and sort of the same tools to assess the flash effect. And in that case, it's what are the properties of these beamlines, right? That are driving the difference that's being observed. Is it the pulses, or how the pulses of radiation are delivered? Whether they are, is it like a continuous radiation delivery, or are these like very short pulses, that are given sequentially? So I mean, again, like there's still a lot of questions, and I mean, I think the flash effect is real, right? There's plenty of evidence that it's real, but yeah, it's still, again, I mean, an interesting question to try to understand really what's behind it, right? So, yeah.
Thank you. That was an amazing talk. So in continuation of the same question, so chemistry-wise, what do you think could be happening, like whether the 40 gray per second that's met by a continuous pulse, or like with, let's say 40 pulses, 1 gray each of like nanosecond or microsecond pulses? Or what do you think could be happening that could be like differentiating between these two?
So you said either 40 gray per second continuous, or like having individual pulses like during a short time, right?
Yeah.
Well, you know, if you look at oxygen beyond a certain, like if you go below a certain pulse width, you know, it becomes almost no difference, because it's all driven by the resupply. And the resupply, you know, if it's below like a fraction of a second, it doesn't really make a difference. However, I think in terms of the very short-lived radicals that are being generated, I think it does matter if you're giving, you know, radiation continuously over, let's say, 100 millisecond, or you have, you know, discrete pulses of like that are about nanosecond, right? Those radicals, they disappear very quickly, right? So the radicals created by the first pulse will be gone by the time you deliver the second pulse, right? So they can't really react with each other. And so, or if you give continuously, you know, there's basically a continuous sort of turnover of the radicals. So yeah, I think it does influence the radiochemistry, like but it's, so I think if you know, if we're looking at what's the difference between these two examples you give, it's mainly, I think, the radicals reaction that will be different. And yeah, I do expect there will be some differences. Yeah, it's,
Thank you.
yeah.
Okay, any other questions?
I don't know on Zoom if anyone,
Yeah, I see Peter there. Do you?
I don't know if we can hear any questions, but yeah.
I do. Thanks, Guillem, and welcome to UCI. It's really great to see that line of research is still going on strong. Just one comment to reoxygenation and to the oxygen effect. I mean, you know, we know from old studies that you need to give a really high dose of radiation fast enough to deplete really oxygen. And so some studies that you know, been done in Charlie Limoli's lab have actually shown that oxygen may not play a role, because they observe the flash effect even with lower doses that are traditionally not been, you know, enough to deplete oxygen. So there was a study with 3 gray tandems for 30 gray for the brain irradiations, and you know, they observed the flash effect despite the low dose. So is oxygen completely unimportant? I don't know, but is that the explanation for that? I don't think so either.
Yeah, I mean, I think, you know, I wanted to maybe bring this back. You know, I guess my belief is that oxygen depletion is often being underestimated, because, and this is, you know, what I'm trying to show here. So the idea is, you know, if you give 10 gray, so you start at 20 macromolar here, oxygen, and then give 10 gray, and then there's depletion of 0.4 macromolar per gray. So therefore you go down to 16 millimeter of mercury, right?
Yeah.
That's like globally on the average on the tissue. But if you go on the right one, what you can see is actually the oxygen depletion near the vessels doesn't change. So the oxygen concentration next to the vessel is the same, right? Because it's, if you're close to a vessel, there's new oxygen that's going to come in continuously, right? And because people are using these oxygen probes that are injected, you know, often IV, these probes may actually tend to be accumulated closer to the vessel, right? Just because that's just the distribution of them, you know, they may not diffuse very far from the vessel. But then you look at further away from the vessel, you can see there's these blue regions where further away from the vessel, where the depletion is actually much larger. And that's because, well, this is where you see the full effect of, you know, there's no time for resupply, right, in this area. So that's one response is I know the measurements in vivo, like the reports, the numbers that are given usually don't seem very big, but still, I mean, the other thing too is any oxygen depletion, even if it's small, you should see a biological effect, right? I mean,
Yeah.
if tumor is hypoxic, you make it more hypoxic. It's not, I don't know. I mean, it's very sensitive, right? Like if you look at the OER curve, it's oxygen enhancement ratio curve. It tends to, if it's, even a small decrease should be biologically meaningful. And then the third thing is, and I think there's a lot of data now that if you modulate oxygen, whether you add more oxygen or you clamping the tumor or clamping the tissue, the flash effect basically either disappear or so the flash effect is very sensitive for some reason to oxygen level. But I mean, I agree. I mean, I think it's I think ideally what we would like to do is, so this picture I have being able to actually make it a measurement instead of a simulation. Because I think maybe, we would see, maybe there is more significant depletion that's going on in these niches that are far away from the vessels. But yeah, I know, so Charles, Charles, I don't know if anybody from his lab is here, maybe, but yeah, I know he's shown flash effect with fractionation, right, for a relatively small dose, 3 gray. Yeah.
Yeah.
Yeah. Thank you, Peter. And I know you're busy today. I'm sorry.
I know, but if you want to come by the clinic, Guillem, that I'm happy to give you a picture of the new thing, of the new hospital. I'm not too far away, but I can't get to the time.
Yeah, I know.
I'm so sorry.
Yeah. But
Thanks, Peter. Any other questions?
Yeah.
Go ahead, Pete.
Thanks for the talk. I have one question regarding to your simulation of 3D brain vasculature, and you mentioned it would be really nice to measure this oxygenation in that context. Do you have any idea when you do the simulation, what's the key factor or indicator that's super important to predict the oxygenation in your model?
I think the diffusion rate is very important, probably, diffusivity.
Right. I mean, those parameters that might be, for example, relevant to imaging that we could observe.
Oh, parameter. So I don't know if I quite understood the question, but I think if we I know there's microscopy techniques that can measure oxygen.
Like such as from the blood flow or?
Yeah, so no, we're trying to measure it away from the blood, right? So we're not looking at like hemoglobin saturation, because we want to know oxygen in the extravascular space. There's no hemoglobin. So typically, we need to use a externally injected probe, like a phosphorescent probe usually, that would diffuse in the extracellular space and then measuring the quenching of the probe, right? That would be, so typically that'll be done with like lifetime, method. So what we did was just a simple lifetime probe, but you could do lifetime microscopy, I think, fluorescence lifetime microscopy. The question is, can you do this and then have this beam of radiation being delivered to your model at the same time? That's much harder, or to your animal, right? So I know there's some methods in vivo to do fluorescence lifetime microscopy, in the brain, for instance, in live mice. But we would like to do it while also delivering like this radiation. So it's a very technically challenging experiment, I think.
Right.
Yeah.
And in your simulation, you will start with that 3D vasculature structure. Is that something important as an initial condition as well?
Yes. So that's used as the boundary condition. The way the simulation is set up is, we have the vascular network, right? And then we kind of simplify things. We don't assume any convection. It's all diffusion. But because oxygen is a very small molecule, so it's mainly diffusion anyway. And then we just set a boundary condition on the blood and say basically blood is at a constant oxygen concentration. And then, it's basically a finite element model where we, model the diffusion, the consumption. Yeah, so it's
right.
Yeah.
It sounds to me like even, the structural imaging of this 3D vasculature could be somehow helpful in your case because you can start with that.
Yeah.
Right.
Yeah. I mean, sorry. Yeah. I don't know if I quite understood that.
Thanks.
Yeah. Thank you.
Any other questions? So I have a question. You talk about those organoids. It's a great model. You don't have to use animal. Maybe you don't need the protocols. But it's kind of a simplified model. So
Yeah.
people might criticize, well, you don't have microenvironment, you don't have immunoresponse, and how do you simulate like the real situation of tumor?
Yeah. I mean, the dream is, people thinking about, well, can you have chips for different organs, connect them all together, and now you have a human on a chip, right?
Oh,
You have kidney, you have spleen, liver. You can have immune cells, I mean, they usually, so these organoids, especially if they are grown in a liquid interface, they preserve the immune cells from the donor. Fibroblast, you can have vessels. I mean, it's the truth that maybe people won't, maybe not people will say is it's actually very expensive to do these tumor on a chip or organ on a chip models. Just the cost of the cells, you need human cells. So human primary cells, right? Where do you get those cells, right? It's very expensive to buy human cells. You have to control, media very carefully. You have to, it's a very complex system. You get sometimes bubbles and things go.
how big they can grow? Can they grow? Like,
Typically a few millimeters, yeah, a few
Mhm.
millimeters in size. Each organ is, yeah, a few millimeter wide. So it's not like doing tissue engineering, trying to like print out a new liver.
covered or
Yeah, for grafting. So other people are working on that, trying to grow large organs for graft, right? For
Yeah.
like to use in patients. So this is more like using model. We keep them small because it's expensive to go bigger. You need more cells if it's bigger, right? But animals are very efficient. It's like a mouse is like a little incubator. You just give it some food and it basically takes care of everything, right? You don't have to, it's very, in some ways, it's much less expensive to do animals, right? Especially mice. Mice are very inexpensive. Their food only costs a few dollars, right? And then they just put them in a cage and they're happy, right? So these are, these could cost like thousands of dollars per experiment. So yeah, it's to do it well is very costly. So, but I think we get human relevant data, right? And so that's kind of the promise is that these are human cells. Yes, it's simplified. Like it's not everything is not exactly the same. But again, the value of a model is not necessarily trying to reproduce every aspect of the human biology. You just want your model to be good enough that it gives you valuable answers. Like that if you have if you ask a question like toxicity of a drug or therapeutic effect of a drug, you get an answer that's useful, right? So it doesn't mean you have to reproduce every aspect of the biology, right? It just has to be, it has to provide value to people who are asking the questions. So
Right. Okay. If no more questions, thank you, Speaker.
yeah, thank you, Rob.